In this video, we'll be looking at part 2 of this chapter. Towards the end of our discussion in part 1, we looked at why nonpolar gases are poorly soluble in water. Similar to nonpolar gases, nonpolar compounds are also poorly soluble in water. And nonpolar compounds force energetically unfavorable changes in the structure of water. When water is mixed with benzene or hexane or 2-methylpropane, which are all hydrophobic organic compounds, neither liquid is soluble in the other, which means that when you add these organic compounds to water, they form a separate layer. And you may have seen this in the case of oil, which happens to form a separate layer as well. Because they're hydrophobic, these compounds are unable to undergo energetically favorable interactions with water molecules, and they interfere with the hydrogen bonding among water molecules. If you can see, 2-methylpropane is made up of carbon and hydrogen, and water has oxygen and hydrogen. As I said before, there is no hydrogen bonding that happens between a CH and an OH or an NH. So this molecule cannot form hydrogen bonds with water molecules. Neither can it interact electrostatically because it doesn't have any charges. All molecules or ions in aqueous solutions interfere with the hydrogen bonding of some water molecules in their immediate vicinity. Now, if you were to consider another molecule like acetone, which is slightly more polar than 2-methylpropane, the only difference is the carbonyl group here, right? But having a carbonyl group adds an oxygen to this molecule. This oxygen can make hydrogen bonds with water molecules. And how can it do it? Because this oxygen can accept hydrogen bonds. For example, it can accept this hydrogen or this hydrogen or this hydrogen, right? But it can only accept 2 hydrogen bonds because it has 2 lone pairs. So what acetone is therefore soluble in water, unlike 2-methylpropane, right? So what I was trying to say here is, even though you add acetone to water, there is always some hydrogen bonding network among water molecules that gets disrupted. Now, if you add polar solutes such as sodium chloride, it is completely different, as we have seen before, because they form electrostatic interactions with sodium ion and chloride ion. Hydrophobic solutes do not offer any compensation the way sodium chloride offers to water, right? We have seen as to why delta G is negative for the dissolution of sodium chloride in water. However, delta G for the dissolution of 2-methylpropane is not negative. And why is that? One of the important reasons is because water molecules in the immediate vicinity of 2-methylpropane, which is a nonpolar solute, is constrained. And their possible orientation is that they are highly ordered structures. This decreases the entropy of the water molecule. So if you decrease the entropy of water molecule, the system, the whole system, which is dictated by delta G equal delta H minus T delta S, the T delta S term does not get negative anymore, right? So delta G does not get negative. And so nonpolar compounds like 2-methylpropane, the dissolution of which is not energetically favorable. Now, In the subsequent sections, we'll see as to how amphipathic molecules are soluble in water and what is the energetics behind it. So, what are amphipathic molecules? Amphipathic molecules contains regions that are polar or charged and regions that are non-polar. Amphipathic compounds will dissolve in water, but the hydrophobic portions will tend to cluster. If non-polar or weakly polar molecules are dissolved in water, energetically unfavorable water structures are formed around the non-polar solute. Also, the clustering of non-polar regions helps to minimize the amount of hydrophobic surface area exposed to polar water molecules. And thus, these hydrophobic interactions among the non-polar regions create structures known as micelles. We'll talk more about these points in the upcoming slides. Let us consider a scenario when a long-chain fatty acid is placed in water. As you can see, a long-chain fatty acid has a hydrophobic tail and a hydrophilic head, which is called as a head group. Because it's a fatty acid, the head group has a carboxylic acid functional group. Now, this hydrophilic head group can interact with water molecules favorably because it can form hydrogen bonds with them. However, the hydrophobic carbon tail is highly nonpolar and cannot interact with water molecules, right? So what happens here? Now, as I said before, water molecules have high entropy because they're flickering clusters of H2O, and they can just form clusters among themselves and break, and they can move freely around themselves, right? So high entropy. But when a long chain fatty acid is immersed in water, the entropy of water molecule that surrounds this hydrophobic region decreases, right? And it becomes highly ordered and form cages around this hydrophobic alkyl chain. So the low entropy is thermodynamically unfavorable, and these hydrophobic solutes have low solubility. But what we are talking here is the case of an amphipathic molecule, like a fatty acid. So how does amphipathic molecules get around this problem of low entropy? So this slide reiterates the scenario that I explained in the previous slide, right? Dissolution of amphipathic lipids in water. Lipid molecules disperse in the solution, and nonpolar tails of lipid molecules are surrounded by ordered water molecules. And the hydrophilic portion of the lipid molecule interact with water molecules. So the entropy of the system decreases because the water molecules that are close to the hydrophobic tail gets ordered, right? So as a result, the system is in an unfavorable state. So to circumvent this problem, nonpolar portions of the amphipathic molecule aggregate so that fewer water molecules are ordered and entropy increases. So instead of one hydrophobic molecule that is distant from the others, they all cluster together so that the number of water molecules around this hydrophobic region is lower, right? And so this makes sure that the entropy of the system does not decrease. All nonpolar groups are sequestered from water, and then released water molecules increase the entropy further. So all the water molecules that were released from this cage-like structure increases the entropy further. Only polar head groups are exposed, as you could see. So when all these hydrophobic molecules come together, there is high enough concentration of these amphipathic molecules to completely aggregate and form micelles. So micelles are these structures where highly ordered structures where hydrophobic regions of an amphipathic molecule come together and interact with each other, whereas hydrophilic portion of these molecules are around them and they interact with water molecules. So to summarize, the nonpolar regions of the molecules cluster together to present the smallest hydrophobic area to aqueous solvent. And the polar regions are arranged to maximize the interaction with the solvent, a phenomenon called as hydrophobic effect. These stable structures of amphipathic compounds then result in micelles, right? Now, micelles may contain hundreds or thousands of molecules. The forces that hold the nonpolar regions of the molecules together are referred to as hydrophobic interactions, and the effect is called as hydrophobic effect. So the hydrophobic effect is really an effect of the hydrophilia of water and changes in entropy. Let us consider the energetics of solubility. So what is the thermodynamics behind solubility of amphipathic molecules? Remember, every biochemical process adheres to this equation, and delta G depends on delta H and delta S at a fixed temperature, even solubility. By forming micelles, the amphipathic molecules are both decreasing delta H via primarily a greater number of water hydrogen bonds and simultaneously increasing and decreasing delta S. The increasing delta S is for water molecules and decreasing delta S is for the micelles, the hydrophobic regions of micelles. However, the increase in delta S for water molecules supersedes the decrease in delta S for the hydrophobic portion of amphipathic molecules, and hence the entropy of the system increases as a result of this phenomenon. Amphipathic molecules are more ordered, but water is more disordered by not forming unfavorable shell around alkyl chains. And finally, the entropy change is often the deciding factor in a reaction's spontaneity. You know this, and this is also the case when it comes to solubility. Hydrophobic interactions also allow formation of micelles, vesicles, and bilayers. Vesicles and bilayers are directly correlated to biological membranes, and we'll talk about these in detail when we look at biological membranes. In addition, binding sites in enzymes and receptors are often hydrophobic. Such sites in enzymes and receptors can bind hydrophobic substrates and ligands such as steroid hormones. Many drugs are designed to take advantage of the hydrophobic effect as well. For example, there are lots of drugs that bind in the active site of enzymes via hydrophobic interactions, and that is how they become better than substrates and inhibit proteins and enzymes. Disruption of ordered water molecules is part of the driving force for binding of a polar substrate to the complementary polar surface of an enzyme. Entropy increases as the enzyme displaces ordered water from the substrate and as the substrate displaces ordered water molecules from the enzyme surface. And as a result, the substrate interacts with the enzyme, and this interaction is energetically favorable because the displacement of water molecules increases entropy, right? In addition to hydrophobic interactions, there are other biomolecular interactions that exist among biomolecules. Dipole interactions is one of them. Dipole interactions are electrostatic interactions between uncharged but polar molecules. Hydrogen bonds are a subset of dipole interactions. Ionic or coulombic interactions are between charged species. These are electrostatic interactions between permanently charged species or between the ion and a permanent dipole. We saw this when sodium chloride was dissolving in water, right? Sodium chloride becomes Na plus and Cl minus, and they have charges, and they make electrostatic interactions. Finally, van der Waals interactions. That happens between all atoms, regardless of polarity, whether it is a polar compound, a non-polar compound, a charged compound. Van der Waals interactions is universal among all these compounds. When in close proximity, the two nuclei induce dipoles in each other and have a weak interaction or a weak attraction. There's a steric repulsive component involved in this as well. Examples for some of these non-covalent interactions that we talked about are shown in this table. First, hydrogen bonds. So we saw as to what hydrogen bonds are, right? Between neutral groups, between peptide bonds. So there's a hydrogen bond acceptor and there is a hydrogen bond donor. Ionic interactions, where there is where there is attraction between a positively charged species and a negatively charged species. In this case, ammonium or the side chain of an amino acid, which is positively charged, can interact with the side chain of another amino acid, which is negatively charged, right? In this case, ammonium ion interacting with a carboxylate ion. And there could be repulsion when there are two positively charged species that are in close contact. Hydrophobic interactions, right? Any two atoms in close proximity that are hydrophobic or non-polar, and they interact with each other so that they reduce the water shell around them and increase the entropy. Van der Waals interactions, any two atoms in close proximity have van der Waals interaction. For example, these two atoms, this oxygen and this oxygen, or this oxygen and this hydrogen, there is always a van der Waals interaction among them. This nitrogen, the hydrogen in this positively charged species with the oxygen of this negatively charged species, there is a van der Waals interaction. And van der Waals interaction between the CH from this molecule and CH from this molecule. So van der Waals interaction is universal, and it is there for two atoms that are in close proximity. It can be attraction or repulsion. Van der Waals interactions, or London dispersion energies, have two components. Attractive force, or London dispersion, depends on the polarizability. Repulsive force, or the steric repulsion, depends on the size of atoms. Repulsion dominates at very short distances. For example, if two atoms are very close to each other, repulsion dominates. Attraction dominates at longer distances, typically 0.4 to 0.7 nanometers. There is a minimum energy distance, which is called as the van der Waals radius, or contact distance. As you can see from this table, each atom has a characteristic van der Waals radius. This is a measure of how close an atom will allow another atom to approach, right? Now, if you look closely the van der Waals radius is slightly larger than the covalent radius for single bond. So which means that this is slightly a larger radius, so there is enough room for a certain atom to come closer. And the bigger the atom, the larger the van der Waals radius, as you can see from the trend here. The question is, what is the biochemical significance of van der Waals interactions? First, it is weak individually, which means it is easily broken and is reversible. To give you an example, the van der Waals energy is only 4 kilojoules as compared to the energy to break a CC single bond, which is about 410 kilojoules. It is universal. It occurs between any two atoms that are near each other. The importance is determines steric complementarity, stabilizes biological macromolecules, for example, stacking interactions in DNA, and facilitates the binding of polarizable ligands. The non-covalent interactions that I described before, such as hydrogen bonding interactions, hydrophobic interactions, dipole-dipole interactions, ionic attraction or interactions, and van der Waals interactions are individually weak. When you compare these interactions individually with covalent interactions, covalent interaction is much stronger, right? About 210 kilojoules per moles. Whereas these non-covalent interactions, such as ionic interaction, is about 4 to 80 kilojoules per moles. Van der Waals forces is between 4 and 11 kilojoules per moles, and hydrogen bonding interaction is between 12 and 29 kilojoules per moles. Biomolecules have a large number of sites to participate in all of these interactions. And when you add all these forces together, they can dramatically impact structure and function. Macromolecules such as proteins contain so many sites of potential hydrogen bonding or ionic, van der Waals or hydrophobic interactions that the cumulative effect of the many small binding forces can be enormous. For example, in case of a macromolecule like a protein, and in this case hemoglobin, which is a very famous protein, and all of us may remember as to what hemoglobin is, right? Now, hemoglobin has, in its most stable conformation, is a homotetramer. And as you can see, it has 2 alpha subunits and 2 beta subunits. The alpha subunits in gray and beta subunits in blue. Now, you do not have to understand right now as to what subunits are, because I will talk to you and I will explain it to you and I will teach you about subunits in chapter 3 and chapter 4, right? Now, the specific example here is to make you understand as to how cumulative effects play a role in protein structure. Right. Now, in this case, right, the most stable structure is usually that in which weak interactions are maximized. The folding of a single polypeptide, in this case, into a 3-dimensional shape like this shown here, right, is determined by the cumulative effects. And the binding of each subunit to one another, for example, the gray subunit to blue subunit, or a blue subunit to another gray subunit, is dictated by these weak interactions. And that is what keeps or holds the protein together. On the right is another structure, the same structure of hemoglobin with water molecules bound in it. Now, remember, these are crystal structures, X-ray crystal structures. And what are X-ray crystal structures? X-ray crystal structures are nothing but structures that are obtained by diffracting a crystal with X-ray, and the electron diffraction patterns can thus be Fourier transformed to obtain three-dimensional information of a molecule, right? Now in this case, we'll talk about this X-ray diffraction in depth in chapter 3 and chapter 4. But to give you a perspective here, water molecules are bound to this protein hemoglobin as shown here, right? Now these bound water molecules can be detected in aqueous solution by other techniques like NMR, nuclear magnetic resonance, which you may be familiar with, right? And they have distinctively different properties from those of the bulk water of the solvent. For example, not osmotically active. These water molecules are not osmotically active. And what is osmosis? We'll talk about in the next part. Now, these tightly bound water molecules are essential for the function of hemoglobin as well. And we'll learn about this later on. But how are these water molecules bound to this protein? That is the crux of the matter in this slide. How are these water molecules bound? The water molecules are bound by weak forces. And one of the examples is hydrogen bonding interaction and van der Waals forces, right? So the take away point from this slide is that cumulative effects, such as the non-bonding interactions, all four of them, hydrogen bonding interaction, ionic interactions, hydrophobic interactions, and van der Waals interactions, right? All four of these are responsible for macromolecular structures.